Okay, the next speaker is Patricia Maurice, who's a professor in the Department of Civil Engineering and Geological Sciences. She's a past director of the Center for Environmental Sciences and Technology here at Notre Dame. In addition to authoring many scholarly articles, she's also written a successful textbook entitled Environmental Surfaces and Interfaces, from the nanoscale to the global scale. Her research interests really span a considerable number of areas, from geochemistry to now geobiology, where I think she's made some nice collaborations with people in other departments, including my own. Her talk has been changed to Environmental Nanogeology, what nanotechnology can learn from nanogeology, and vice versa.
Thank you, Paul. Don't want to pull a politician here. Yes, I decided to change my title after I saw where I was placed and the fact that a lot of preliminary things on nanotechnology would have already been addressed. And so since I am partly a nano engineer and partly a nanogeologist, I thought I'd wear the geology hat today. This is Notre Dame, so I can say this. God made nanoparticles right from day one. And they're out there, and nanogeologists, short people like myself, as well as tall geologists, have been working on nanoparticles for a very long time, for many decades. They're out there, they're what we study. And I see some things that we've known for a long time that we can help teach the nanotechnology community, and also things that the nanotechnology is learning that's really helping nanogeologists to better understand the world. So I thought that would make a better talk at this time. There are a couple of things I'd really like to accomplish with this talk. First off, what is the nano world like there, the natural world like out there from the nano perspective? The second thing is that I'm going to go back to real basics, and I apologize because I know I'm going to bore certain people, some people in the audience. But we're using terms like surface area, and we've seen this picture over and over again about how surface area changes with particle size. Why does that matter? Why does that matter from a very microscopic view as well as a more macroscopic view? What's happening when we change surface area? What's happening when we change particle size? And specifically, I'm going to focus on geological materials, which have different things going on in them than some of the biomedical materials, although I was really very interested to see that there already was some nanogeology in this conference. Magnetite is a geologic material, and a lot of these other materials, the silver, that's all a very geological question as well. So with that, here we go. All right. Again, I'm going to repeat a lot of things, but from this geological perspective. So it's important to our understanding of the Earth as geologists to investigate not just the global scale. Right now, there's so much funding and so much, real hype, and I shouldn't say hype because it is meaningful, about global change and how the global system works. But we also have to understand nanoparticles and really the difference in scale between us and the Earth as a whole, especially looking at from outer space, for example, and us and a nanoparticle is about the same difference in scale. So we're kind of sandwiched right in the middle between these. Nanoparticles definitely have a strong impact on even global change. What are some natural nanostructures that are out there? This picture, I apologize, it's probably not too easy to see it. This is some of the first nanoparticles that I worked on. I started working on them in the 1980s. This is Humic Materials. When you have a soil, that nice brown, dark soil, that's this organic matter that comes naturally from the breakdown of plant debris in soils. It passes into the soils, and then it ends up passing as dissolved humic materials into the underground waters and into streams. And it's very important environmentally because as go the humic materials, so go a lot of the pollutants. They bind metals, they bind organic pollutants, etc. Whether they adsorb to a surface or pass into the solution really affects that mobility. What you're seeing here is nanoparticles, nanoaggregates of natural organic matter imaged with AFM in solution, where you end up with these ring structures. So the individual nanoparticles of humic substances are only about a nanometer in size or so. but depending upon what surface they're on when they adsorb or what solution they are in, they can self-assemble into different types of structures. In this particular case, they're self-assembling into these rings of multiple molecules together. And we believe that this ring structure is important because the little domains in the middle, it has little micro rings inside of it. It's like fractal. Those little tiny, tiny little rings inside of it may be playing a role in the partitioning of hydrophobic molecules, for example, into the really small little ones that you really can't see too well, and then you've got these larger ones as well. You can kind of see, I'm sorry, this image just doesn't show it very well, little tiny rings within rings. So humic substances, which are out there everywhere, are themselves nanoparticles, and they self-assemble in different ways depending upon what surface they adsorb to. Clays, we talked yesterday, Maria mentioned the swelling clay marlinite, which Maria and I have the distinction of probably the only people in the history of this university who've broken each other's. She broke my tooth and I broke her ribs in karate. So we're very good at sparring over things together. We're good sparring. Yes, I know. We're strong, but we're small, but we're powerful. But at any rate, swelling clays have interlayers that are, just at the border between angstroms and nanometer scale. And as they swell, they can take molecules into them, and the conformations and reactivities of those molecules can change in the interlayer. So, for example, you may have an organic molecule that has a very complex three-dimensional structure out in water, but if it gets into that interlayer, it may take on, for example, a more planar conformation that really changes its reactivity. So this is a nanoscale, angstrom to nanoscale space where you've got different properties than bigger or smaller. We have something we call nano minerals. This is the nano mineral ferrihydrite. It's an iron hydroxide that often is precipitated by bacteria or found attached to bacteria. This ferrihydrite, it's very difficult to study. It can have different amounts of reflections in X-ray diffraction, for example. Its properties, its structure can change depending upon how hydrated it is. So if you dry the sample and try to analyze it, you may get a different result from if it's wet. Has really high surface area. It can really affect bioavailability of anything that's adsorbed to it, as well as it itself as a source of iron. We had a little bit of talk on magnetite this morning. This is natural magnetite that's formed in this case, this is about 50 nanometers, if I'm reading this correctly. These are little magnetite particles that have formed inside the bacterial cell wall. And these bacteria are known as magnetotactic bacteria. They precipitate, bioprecipitate magnetite right in their cell wall, and that allows them to align themselves relative to the Earth's magnetic field. And that can be very useful to them in how they're interacting environmentally. Bacteria also, this dissimilatory iron-reducing bacteria, dissimilatory metal-reducing bacteria, can have coming out of them something called nanowires. These nanowires are little materials, little wires that are exuded out of the bacteria that can transfer electrons, and you can end up precipitating various minerals or dissolving various minerals that the nanowires come in contact with because of this electron transfer. So here you've got the bacteria with these nanowires coming out from it, where you've got little iron oxides that are precipitated, in this case magnetite, because of this electron transfer. It ends up that these bacteria also can send out these nanowires and dissolve iron oxides to use for various purposes. So they can both precipitate and dissolve. Nanoparticles are very important to a lot of issues related to contaminant fate and transport. Here we have, this is our work. Someone this morning mentioned Mike Ocella. Mike Ocella was one of my advisors when I was a graduate student at Stanford. He went on to move to Virginia Tech kind of in the middle of my program. But he's been doing a lot of work at what's now the largest Superfund site, the Clarks Fork River. One of these sad legacies of mining, of course, basically I'm a firm believer in geology because everything in this room somehow either was grown on rocks or comes from rocks or something, okay? And we've got rocks all over us, minerals, our teeth, everything. But one of the sad legacies of mining is that you have a lot of mine waste, where as the water percolates through that mine waste, it becomes very acidic, It picks up metals, and you end up with massive contamination in certain sites. Keeps people like me in business. You know, my students can get really good jobs, and it's going to keep going on for a very long time, but we really don't want to see it happening. At this Clarks Fork Basin site, what he's found to the Clarks Fork River is that what's really controlling the contaminant transport is nanoparticles of iron and manganese oxides. These iron and manganese oxides precipitate in this environment, and one of the things they do is they can just transport down the river themselves, or they can attach to different surface site features on other grains, and then you end up with a large particle that has a lot of nanoparticles on it. And those nanoparticles have very high surface area. I'm going to talk about why surface area matters in this, and they carry a lot of the pollutants. And because they're nanoparticles, they react very quickly, and that means the pollutants that are attached to them can really change very quickly. So for example, you can have photocatalyzed reduction of the iron oxides that kicks the metal, the toxic metal, off, and then at night, or when it gets a little deeper, you can have the opposite process. So you can have very fast reactions when you've got these nanoparticles in high surface areas. So these nanoparticles are really controlling the fate and transport. Here's another, global climate change. What do nanoparticles have to do with global climate change? Well, I haven't even shown here the fact that nanoparticles in the atmosphere can play a large role in radiative forcing and how the solar energy is absorbed or emitted, etc. Here's something, black carbon. We heard about carbon black. There's also black carbon. Black carbon is released by burning of fossil fuels, by forest fires. These little nanoparticles of carbon, they can be different sizes. They can have different structures depending upon how they were formed. They transport through the environment. For example, here's a NASA picture showing black carbon that's formed from a massive forest fire in Alaska. They can transport a lot. One of the things that happens because they can transport so far, they're small, if they get up into the upper atmosphere, they can move a lot, is they are changing the albedo of ice in Antarctica. I've worked with Diane McKnight on some research looking at nanoparticles and microbes in all Antarctica. This is one of her pictures. When these nanoparticles of black carbon, and carbon black probably too, deposit on the glaciers, they actually make the glaciers darker colored. And so the amount of light that's reflected off the glaciers decreases, and you get more melting of the glaciers, which is increasing the feedback loop to cause, the less glacier you have, the less albedo, the more solar energy is absorbed instead of reflected, and it's changing light. Now you would think, oh, this must be minor. This is actually getting to be quite major, and it's a big area of research now in climate change. A lot of this black carbon that we're finding on Arctic and Antarctic glaciers, we can directly trace to burning of coal in China. So, and it is an accelerant of climate change. All nanoparticles. Soils are the ultimate nano environments. All of our food comes from soils, ultimately. And so, we worry a lot about water. We worry a lot about climate change. We worry a lot about all sorts of things. Not enough people really focus on soils and how destruction of soils and soil loss and destruction of the quality of soils can be affecting our food and our water. In soils, which this is a typical soil profile that you may see in this case in New Jersey, you've got the organic matter up on the top. It gets degraded and goes through, and it kind of leaches things. This is something called the pod soil. And then down in here, you've got all sorts of nanoparticles forming. So here, for example, this is a TEM image. Sorry, I don't have a scale on here, of clays and iron oxides that are forming at very, very high resolution in some of these pores. Here are some of those particles. Here we see a bacteria that's probably actively bioprecipitating material on it. Sometimes it could be happening as a passive process. So, this is an environment. Soil scientists have been nanotechnologists, nanoscientists, for as long as soil science has existed. And your farmer really has to be concerned with nanoscience, nanotechnology naturally, and then what's happening when we get engineered particles in there. The first clay minerals working group was first started in the 1940s, around the time of the Second World War, And then the Clay Mineral Society was founded in the 1960s. And I want to make this point. It was to recognize that very small minerals, very small particles of minerals, have unique properties. And so we needed a separate society to be able to study them. And perhaps even more importantly, they are extremely difficult to study. So someone who typically does X-ray diffraction to get the crystal structure of a mineral, that's a pretty straightforward thing to do. When you get to these little nanominerals or micron scale minerals that you have with clays, you may have a montmorillonite clay that's an interlayering of montmorillonite with a whole different clay illite. And it's very hard to tease that out. So we need a whole different scientific group in order to study it. This is kaolinite clay up here, for example. Whoops, wrong button. I knew I was going to do that. This one's about 300 nanometers across. There's another particle here that's probably about 100 nanometers. This one here is about 700 nanometers across. And you can get down a broad size range that scales the nanoscale and on up. Usually they tend to be quite thin, which definitely makes them nanoscale in terms of their thickness. And I want to point out they can have all sorts of features on them. Like, for example, these are some steps I'm going to talk about later on. Any of you in here who've ever done atomic force microscopy, when I was a student at Stanford, I opened up the first one that was ever sold by Digital Instruments. And they didn't have any way of calibrating the height that you were measuring with the AFM. And we ended up working very closely with them that we knew what the height, the spacings of steps should be on this clay and on mica. And we were able to use that for them to calibrate the heights. So it was the mineral nanoparticles and nanoscale structures on minerals that allowed AFM to calibrate its first height range down at single angstroms or a few angstroms. Clays are ubiquitous. They're found in all sorts of cosmetics, masks, all those types of things. Pure minerals that women put on cosmetics, that's all clay. A lot of it's clays. They've, of course, been used in porcelain. They're used in automobiles, in all sorts of parts in automobiles, you name it. And so we have actually a lot of knowledge about how certain types of nanoparticles interact out in nature and interact with each other because we've been following this for a very long time. Better speed this up a little bit. Okay, so we've been talking a lot about definitions, and I wanted to kind of go through what the geologic community is doing along those lines. Hocella et al. In 2008, this was a paper that came out in Science. I was a co-author, and I fought this every step of the way. And then somehow he slipped it by me at the end. But he was my former advisor, so he can get away with that. They're as small as roughly 1 nanometer and as large as several tens of nanometers. And this is where I won, in at least one dimension. So we were fighting over, 100 nanometers, whatever, the nanonin, all right, in at least one dimension. So now this includes the clays, for sure. It does, it's kind of difficult. Banfield and Zhang in 2001 pointed out something we've talked about before. Nanoparticles might be defined based on the size at which fundamental properties differ from those of corresponding bulk material. The size range that constitutes a nanoparticle may be somewhat different for different materials. We've talked about that. The geologists have been struggling with it. We've seen this kind of in a different format. Let's show it at how an earth scientist would look at nanoparticle size. So here we've got the sizes of different particles in nature. An angstrom is about the size of a bond or several bonds, depending upon exactly what you're talking about. But that's about the general scale it is. Nanoparticle, if you look just linearly, is about 10 of that. So ultrafine particles are about 0.1 micron. Dissolved versus particulate, 0.45 micron. Molecules, nanoparticles, colloids, viruses, all tend to be in the dissolved or at the boundary with the particulate. I put this little thing out here because you can potentially have a very long nanorod. So that's the idea here. Viruses, bacteria, and sands. Bacteria can have features that are nanoscale features, but they're not nanoparticles. Nanoparticles are often sampled in the dissolved fraction, so much of what we've done on environmental chemistry of natural waters, we haven't even been considering the fact that a lot of what we're probably working with is nanoparticulate. Defining nanominerals, you've gone through various definitions. I wanted to go like how geologists are doing this. A mineral is I'm going to read this because the wording has been fought over for many, many years, and now we're fighting over it even more. It's a naturally occurring, So if you got an emerald ring that wasn't naturally made, it's not a mineral. Okay? Naturally occurring, homogeneous, so it's got the same structure composition relatively, inorganic, solid substance, having a definite chemical composition and characteristic crystalline structure. But the nanoscale, I'm going to show you how composition and structure can change as a function of size. So if we've got the mineral hematite, everybody knows the mineral hematite if you work in my field, okay? And if you try to apply this definition to it, when you get down to nanoscale size, its structure and composition change with size. It's not homogeneous, it's not the same. It's still hematite, but is it really? Does it fit the definition of a mineral or not? Also, many nanominerals, or mineral nanoparticles, are biologically precipitated, and the term inorganic has often been taught to mean that it also has to be formed by an inorganic process. So we're really changing potentially what we're going to have as a definition of mineral as we look more at the nanoscale. Nanominerals are defined as minerals such as the mineral perihydrite that only exist in the nanoparticle size range. You don't find them bigger. Mineral nanoparticles may also exist at larger sizes. So you can have a nanoparticle of zinc sulfide that's the same thing as bigger, but it just happens to be nano size. Now, I really want to show this type of slide because we have some discussions of how do you define things. The way the geologists are doing it is they're going to say, we're going to define things differently if it only occurs at the nanoscale versus if it's simply a nano phenomenon that we see at the larger scale as well in terms of the material. The fact that nanominerals and mineral nanoparticles can have properties, including stability and reactivity, that change as a function of size, make them fundamentally distinct from the bulk minerals. Okay, I only want to point, this is all this discussion of how size matters and all, and I want to point to the very bottom. In most cases, it is not known below what size these properties will begin to vary for which minerals, because to date, few measurements of this type have been made. And that's the case in so much of the nano world, that we talk about how do you define nanoparticles, nanominerals, on their size or their reactivity or their structure. We really don't have that many measurements of how things change as a function of size. One thing that the geologists have really been focusing a lot on, which we haven't discussed as much here, just a little bit, is what's the lower size limit of a nanomineral. So there are zinc sulfide minerals, such as sphalerite, for example. We know that in water, you can have zinc sulfide clusters that have been measured to be about 0.7 nanometers in size. The unit, the unit cell size for when these minerals is about 0.54 nanometers. In order for something to be called a mineral, it has to have a repeating structure. So you have to have more than one unit cell. So the size has to be somewhere, it has to be bigger than this and probably bigger than this. Zinc sulfide nanominerals as small as 1.5 nanometers have been observed and characterized, and we know they have a crystalline structure. So what this tells us is that the lower size limit for something to be a mineral would be called nano is probably about 1 nanometer. What makes a nanoparticle different? We've been banding this around a lot. I want to give you some very specific examples. At the nanoscale, materials often display these unique properties, different crystalline order or structure, stability and reactivity, thermal, electrical, optical, tensile strength, magnetic properties than the larger molecules. Let me show you some examples of why. This is a model of titanium dioxide particle. Titanium dioxide is widely used. It is a natural geologic material, but it's also used as nanoparticles. We saw some pictures yesterday. Surface area itself is important, but also how much surface you have relative to the bulk can really change reactivity. When you get to small titanium oxide particles or any mineral particle, you get a larger proportion of atoms associated with the surface. The atoms that are associated with the surface are actually bonded differently from how they are in the bulk. So that means that when you have a large particle, you've got this surface that's bonded differently from the bulk, and it's going to change its properties, its reactivity. But when you have a small particle, that a very high percentage of the total particle is surface, you've got a lot of different bonding than in the bulk. And that's why surface area matters more than just surface area alone. When you look at the free energy of a particle, what controls its stability? The particle free energy can be broken down into the bulk free energy that's related to what mineral it is, for example, and how stable it is relative to other things. But it also has a component of the surface. The free energy associated with the surface is different from that of the bulk. So the total free energy is these two combined. When you've got a very, very small particle, you can suddenly get the surface free energy dominating over the bulk free energy. And so the whole stability of that particle can now be dominated by that surface free energy, and it can behave totally different in terms of reactivity, in terms of its stability versus the larger material. Now, we've known for a very long time in dealing with surfaces that if you take a mineral, for example, this is a model, I'll go through it as a bulk structure, you can figure out very nicely exactly the coordination, where the atoms are, how they're relative to each other, what the bond lengths are, bond distances, and everything for a bulk mineral. But when you break that mineral off and it's now a surface, you've removed half of the atoms that things were bonded to. And so you now have all these dangling bonds. If you're in a vacuum, and you do this in a vacuum, they may have nothing to bond with. If you do it in air, they're going to immediately be picking up waters of hydration and hydroxides from the air. Actually, there's some discussion of whether a vacuum really doesn't have the waters of hydration in it also, but theoretically. So here is what this was done by Gordon Brown and some other group people at Stanford, where they looked at the structure of the aluminum oxide. And so this is just a structural model. You've got your aluminums and you've got your oxygens, and you give the angstroms and spacing as you go on down. So this would be an idealized, what does the structure looks like idealized. Now let's actually take the surface of that aluminum hydroxide, like a centimeter scale crystal, and have it with water adsorbed to it, and now do measurements with a synchrotron of what those bond distances and bond lengths and everything are in that situation, actually at the surface in a hydrated environment. And you see the distances, this would be water, and then this is the molecule, it's the mineral itself. The distances can be in some cases a little greater or a little bit less between them than in what you would see from just the bulk crystal. So you've actually had some expansion and some contraction, some relaxation and non-relaxation, and that can penetrate down angstroms, at least angstroms, as much as a nanometer or more, depending upon the material. So what this tells us is that the actual bonding environment at a surface is different from in the bulk. Now, this is for a single crystal like centimeter scale. But when you've got a nanoparticle down at 2 nanometers, for example, as I mentioned before, basically the whole particle is surface perturbed. It's going to be different binding than in the bulk mineral, and that's going to affect its reactivity. This is the mineral hematite, the same type of thing, near and dear to my heart. I did my dissertation on hematite, and in fact on this process we're going to talk about here. But hematite is an iron oxide. It's widely disseminated. It's one of the things that gives soils their red color a lot of times that you see, an iron oxide. The bulk structure has this model is really iron atoms surrounded by oxygen, and he's kind of done a model that I have a hard time looking at, except that as a geologist, but not if I'm looking at it otherwise. So you've got, here's what the surface structure looks like if you just were looking at the bulk structure. These really relate to irons with oxygens on them. At the surface of hematite, kind of every other iron atom is missing. It's not there. So what that means is that you've got a totally different coordination environment from if you've got the bulk hematite. And in fact, hematite takes on a more iron hydroxide rather than iron oxide structure at its surface. This is profoundly important for how this mineral interacts in nature and probably how iron oxides interact in the human body. So you've got, under coordination, you've got a more hydroxide-like, and when you get down to a very small nanoparticle, is it hematite, or is it goethite, or is it something in between? What exactly is it? My group has been working quite a bit on this with Maria. In fact, we've been using her XPS. These are some results where we've got XAFs, which is a synchrotron radiation technique, and X-ray photoelectron spectroscopy results on bulk goethite, bulk hematite, which just means large particles of goethite, large particles of hematite. And then what happens when we have hematite that to an X-ray diffractometer is hematite. The X-ray diffractometer says we don't see anything else but hematite here. And we get it very, very small, only 8 nanometers or 3 nanometers. What we see from the XAFs is that as we get very small, so we go from a large crystal of hematite, these are probably about 100 nanometers, to the 8 nanometer to the 3 nanometers, the structure takes on an increasingly more goethite-like character. You can see that in this area. You can see it over in here. And with X-ray photoelectron spectroscopy, which is a surface-sensitive technique, we also see a more goethite-like. Now, it may not be exactly goethite, but the thing is it's more hydroxide, iron hydroxide rather than iron oxide character. So based on the bulk method, which is the XAFs method, which is measuring everything, when you get to the smallest particles, you've got X-ray diffraction says it's hematite, but XAF says that a third of the structure is hydroxide, not oxide. And then when you get to the larger particles, they're all oxide. Now, there may be some hydroxide at the surface, but it's such a small proportion you're not picking it up. With the XPS, you do see that. You see that at the surface itself, that you've got this hydroxide surface, and then as you get to the smaller and smaller particles, it's got a more hydroxide character. So again, this is profound impact on how things react. Because even though if you measure it by X-ray diffraction, which is the technique everyone has used since geology existed and an X-ray diffractometer was made to say what minerals are present here, it only picks up the hematite. But in fact, these tiny, tiny particles have a more hydroxide structure, especially at their surface. Why does this matter? Well, one reason why it matters is because it changes the stabilities of the particles as they go down in size. So there was, when I was a young geologist, I remember going to conferences and people talking about the relative stability of the mineral hematite, maghemite, goethite. These are all iron oxides and iron hydroxides. Magnetite's a little different because it's got some iron too in it as well, which maghemite can have. But and people were really arguing over, well, how come you got this relative stability? Well, our lab does better thermodynamics than your lab does. Actually, it was all just a function of the size of the particles they were working with. That as you go down in size, this is some work done by the really great thermodynamicist Alex Novatsky. As you go down in particle size, which means greater surface area, and you look at the energy associated with transformations, they cross over for the different minerals as you go to smaller and smaller size. So how they are stable relative to each other changes as a function of particle size related to this surface free energy component becoming more important relative to the bulk, related to the change in structure, maybe related to some other things too that we need to further consider. Environmental reactivity of surfaces, I'm going to say of minerals, but of other things as well if they're solids. A lot of times how they react is not based on how much total surface you have, but the density of certain reactive surface sites. So very typically, for example, let's say that this is a flat, perfect mineral, but it's got little steps on it that are maybe monatomic steps or something like that. And then it may have a kink. It may have a vacancy, which would be a hole down in there. It may have an add atom. These are the things that often are controlling how it absorbs a toxic organic or a toxic metal. Okay? And when you get down to very small particles, the density, and this is kind of a bad schematic, the density of those reactive sites really increases. And that's going to affect their reactivity. So here's one example of how this actually plays out in an experiment. Here we dissolved nanoparticles and larger particles, although you may not call all of them nanoparticles, of the iron oxide hematite in an organic ligand that dissolves the iron out of this. And here we have the time and the rate of iron removal. For the larger particle, the rate is about an order of magnitude slower than these little nanoparticles. So this different reactivity really plays out. They dissolve about an order of magnitude faster. Now that's important that when we did this experiment, we did the experiment with the same amount of surface area per particle. And one of the things I really want to stress in this conference is that when you look out there at anything that anyone in my field is doing, a lot of times they're saying this is how nanoparticles react differently from slightly larger particles. But they're not normalizing for surface area in the experiment. They're correcting for differences in surface area afterwards. And they're seeing results that often are artifacts. Adsorption. Here we've got the same particles, how they absorb lead as a function of pH. A little different for the nanoparticles versus the larger particles. Nanoparticles, it's often thought that very small particles are not very stable, and they will dissolve and ripen into larger particles over time. It ends up nanoparticles don't necessarily follow this. Instead, they grow by something called oriented attachment, where here little rhombohedral particles are self-organizing into hexagons. And so you're able to go from a very small particle to a larger particle by an entirely different route. I'm almost done here, just a little bit. This is one of the posters that's out there, some work we've been doing with Jen Dubois' group here, where we've been growing bacteria that need iron in order to grow. They need iron for the basic metabolism in the presence of hematite of different particle sizes. We've used a wild type that exudes this organic siderophore and a mutant that doesn't exude it, so it doesn't have that mechanism to acquire iron. I showed you the dissolution there. This is the growth, the microbial population growth. These bacteria can only grow if they acquire iron. Grown on 72 nanometers, it takes them a long time to start growing, although eventually they do grow pretty well. If you've got nanoparticles, they start growing much, much better. And they grow a little differently depending upon whether you normalize the experiment to the same mass or the same surface area. Over here, we grew the mutant, which can't produce this siderophore. The larger particles that can't get iron out, they can't grow. If you normalize the surface area, they can grow a little bit. If you normalize to mass, they can grow tremendously. These bacteria can use a totally different mechanism for getting iron out of tiny nanoparticles than they can out of the larger particles. And the experiment gives different results, whether you add the same mass, which is different surface area, or you add the same surface area. Some of this is that these nanoparticles actually can attach to the cell wall and maybe even get into the cell wall, and we know that there's a cell wall associated reductant. I'm going to run over maybe 2 or 3 minutes. Sorry. Another big question is that how do nanoparticles move through the environment? Surfaces in nature are charged. They attract negative ions. If it's a positive charged surface, repel positive ions, or vice versa. All of our theory for how a little particle moving through porous media might attach to that surface or not is based on a theory that says that the force here at which we're acting, that the particle is bigger than that force. But nanoparticles are smaller than the distance over which the forces operate. We don't have a theory for dealing with how particles attach to each other when the particle is smaller than the distance over which the force operates. We don't know. Small particles can take very tortuous routes through little pores. Big particles can go only through the larger ones. They can't go through those little pores. That can affect how things will travel in the environment. So if you look at a septic tank leaking, the solutes often will go through a certain distance because, maybe not solutes, but really, really small particles, they go through a tortuous route. Large things like microbes or colloids take only the larger paths and they can move a lot faster. Nanoparticles may move slower or faster depending upon exactly where they can fit in the pores and also whether they aggregate or not. Because if they aggregate, they're now behaving like the larger particles, or they may be clogging pores. So we don't necessarily know how nanoparticles will move in the subsurface. So why does all this matter? Well, we brought up the point that we may need a new definition for what's truly dissolved. Nanoparticles currently are included in dissolved. Maybe we shouldn't be doing that. But can we distinguish nanoparticles from dissolved because you can't regulate it if you can't measure it. This is an example of the EPA recommended water quality criteria for some of the different metal contaminants. Note that copper here, the freshwater criteria is not based on just a concentration, but on something called the biotic ligand model, where you have to measure other concentrations of other things in solution because we know that that affects how they behave in toxicity, for example. Well, part of this is probably also because that some of that copper out there is nanoparticles. So we already are recognizing that different materials can behave differently and that you may need to have different standards. Copper nanoparticles, in fact, have different toxicity than dissolved copper. Here's some zebrafish gills, a control. The nanocopper nanoparticles actually can get in there and disrupt them. They can be quite toxic, and their toxicity is far more than dissolved copper. And you can consider the dissolved copper in this. So let's go on to kind of one last thing I wanted to talk about, one last point. This issue of surface area. We've mentioned it so many times, but it's not usually considered correctly in experiment after experiment. Here is an example where my student ran an experiment of lead adsorbed to hematite at different particle sizes. I already showed you this data. And it shows that the 40 nanometer particles absorb a little bit differently from the 3.6 and 8.6 nanometer particles. So size has an effect. We ran this experiment normalized to take account for the differences in surface area. Usually when people run experiments, they put the same number of grams of the materials in. For nanoparticles that have huge differences in surface areas, that makes a big difference. This is what the results look like if you normalize to geometric surface area. This is what the data look like if you normalize to the mass of the particle. The effects of the nanoparticle size become completely different depending upon how you normalize your experiment, because surface area changes so drastically with particle size when you're small, and you're running a different experiment if you're not normalizing to surface area but to mass. So with that, I would like to conclude by thanking all of my great friends and colleagues who helped to focus on the really small nanoparticles. And I would be happy with the plug for my book to take any questions. Although maybe not too much because I ran over a little bit. Do you have time for a question, or do we just want to move on?
We can take a couple of specific questions for Patricia. Well, I do have one, and that is that you showed the bioavailability being related to the size of the particles,
Yes.
correct?
Yes.
So is there any correlation to the size of some of these ion complexes that you see as storage in bacteria?
That's like the next question that Jen and I want to get into, is that, and also the question of ferritin.
Because one would presume that over evolution you've optimized things so
to get the right size, right.
System.
Right. And of course, the structure changes. One of the things that Jen and I are getting into right now, because it's this biomedical community in here, is that aluminum can play a role in iron deficiency and anemia. And what is the role? How does aluminum do that? How does it play that role? My group has been working for years on how a little bit of aluminum incorporation into iron oxide nanoparticles can completely change how they dissolve relative to organic ligands in soils. And in fact, just a little bit of aluminum can totally poison the ability for them to dissolve in certain soil environments. On the other hand, sometimes it can enhance how they dissolve. So it may be that if a little bit of that aluminum incorporates into some of these iron particles that can be present in microbes or in the human body or whatever, they can really poison how it behaves with respect to how you get the iron out of that. So that's kind of our next area that we're moving in this.